Okay. What we have learned in previous lessons, okay, we are using the adult hemoglobins, okay, adult hemoglobins. So now we're going to look at this. What if we compare these adult hemoglobins with fetal hemoglobin? Okay, so if you look at the fetal hemoglobins, you can see that, or we label them as HbF. So in adult hemoglobin will be HbA, normal. So HbA will be alpha 2, beta 2. Correct or not? We have 2 alpha and 2 beta chains. So we call it alpha 2, beta 2, HbA. If a sickle cell anemia, sickle cell patients, it will be HbS. We call HbS because sickle cell hemoglobin in this case, so it's alpha 2, beta 2, but mutation take place. A mutation in beta chain. Okay, then we talk about fetal hemoglobin will be HbF. F stands for fetal. So you can see that the productions of the fetal hemoglobins or fetal red blood cell actually at the liver first, correct or not? After that, only change to bone marrow. So at the liver, the productions of this fetal hemoglobin HbF, so they are having same 4 polypeptide chains, but 2 alpha, 2 gamma. Okay. So therefore, means that the chain with this gamma, okay, from beta to gamma in this case, actually we change from beta. We change to the beta from gamma. But in this case, in the we call about fetal hemoglobin is 2 gamma. So therefore, the oxygen dissociations will slightly different. Okay. Now, be careful when you use the word shifted or lies or located, because shifted means that they are the same. They must be same hemoglobin, only can do shifted. Are you clear? We can shift it to the right. But this one, there are 2 different kind of hemoglobin. If you look at this graph here, okay, you can see that fetal hemoglobin, adult hemoglobins, 2 different form of hemoglobin, they cannot shift. Are you clear? Shifting, okay, or the shift of the curve only apply when we use the same hemoglobin. Like for example, in the Bohr shift and 2,3-BPG, we add other chemical compound, so therefore the graph shifted. But in this case, it's fetal hemoglobin and adult hemoglobin, we use the words actually located. So the graph or the curve for fetal hemoglobin located at the left-hand side of the adult hemoglobin, or lies to the left. So again, to the right, low affinity, to the left, higher affinity. This is very, very important. Okay, so why? Because in the placenta, okay, in the placenta, O2 have to be released by the adult hemoglobin so that fetal hemoglobin will extract or bind. So if the fetal hemoglobin, they do not have the high affinity, so adult hemoglobins won't be able to release the O2 for the fetus. So mean that fetus won't get the oxygens through this placenta exchange. Okay, so fetal hemoglobins, okay, during the last 7 months of development in the uterus and newly born until roughly a 6-month old. So during this period of times, they have this fetal hemoglobin. So this fetal hemoglobin, you can see that I use the word highlight. I don't say shifted to the left, but I use the word lies to the left or located to the left. So to the left basically means that higher affinity. Why must be higher in this case? Because a developing fetus only can receive the blood, receive the oxygen across the placenta from the maternal hemoglobin. Are you clear? So this maternal hemoglobin means adult hemoglobins. Okay, so the maternal blood has to supply her whole body as well as the fetus needs. So therefore, by the time blood reaches the placenta, the partial pressure oxygen is relatively low. So with low, means the supply of the oxygens to the placenta, so means that fetus have to develop the ability to extract O2 from the mother blood. So means that the fetal hemoglobin therefore must have high affinity for O2 compared to the adult hemoglobins. Are you clear? So you can look at this. At each positions, let's say about like 2.5 or 2, you can see that the percentage saturations for adult hemoglobin is lower than the percentage saturations for the fetal hemoglobin. So means what? Means that in the placenta, adult hemoglobin HbA what they do? They release O2. So how about fetal hemoglobin? Because they have higher affinity, right? They have higher affinity, so they're going to take in. So they're going to bind to O2. So you can see that this is how fetal hemoglobin extracts the oxygen from the adult hemoglobins. Okay, clear or not so far? So not much thing, I mean, for us to actually go into the detail. So understand what you need to know. Very, very simple. So summary here for HbF. So the curve located or lies at the left. So at the left means increased affinity. Okay, so why increase affinity? Because they have to extract O2 from adult hemoglobins, okay, from adult hemoglobin. So therefore, they have to have the higher affinity. Okay, now the second one, we're going to look at these myoglobins. Myoglobin, actually, they are tertiary protein or tertiary globular protein. So it means that they are only have one polypeptide chains. Okay, so they are actually similar to the hemoglobin, but this myoglobin only can be found in the muscle tissues. Are you clear? So myoglobin is a primary, okay, oxygen carrying pigments found in the muscle tissues of the vertebrates in general and almost all mammals. You can see that. Okay, and this myoglobin actually particularly high in cardiac muscle. Okay, myoglobin particularly high in cardiac muscle. So high concentration of myoglobin in the muscle cell allow organisms to hold their breath for longer period of time. So some diving mammal, such as the whale, the seal, you can see that the muscle particularly high abundance of the myoglobin. Okay, also this myoglobin also contains the pigments, okay, the hemoglobins. Contain the heme groups. Okay, myoglobin contains the heme group, not hemoglobin. Myoglobin contains the heme groups, which are pigment responsible for the color of the red meat. Okay, so myoglobins right now, they only consist of one polypeptide chain. Okay, so it means that myoglobin only up to tertiary structure, not quaternary. Okay, so look at the graph again. This is not a sigmoid graph anymore. Can you see that? It's no longer sigmoid, it's not X-shaped. Okay, it's more to what we call the parabolics. Okay, look at this. Okay, so this what actually we can see that. Now look at this. Dissociation curve of the hemoglobin against lies to the left, okay, as compared to the adult hemoglobin. So means that to the left, it must have higher affinity to the oxygen as compared to normal hemoglobin. Okay, so what actually happened? Myoglobin, what is their role? Their role as the O2 storage. We have learned about this myoglobin in the respiration during the oxygen deficit. When the O2 is not enough, when O2 supply by the O2 supply by our body is not enough, okay, then you're going to trigger the release of the myoglobins. Okay, so look at how we analyze this. If you look at about 2 kilopascal here in the muscle, 2 kilopascal, what actually happened? Now look at this. So if you look at 2 kilopascal, for normal hemoglobin or adult hemoglobin, you can see that the percentage saturation is low. So it means that hemoglobin, adult hemoglobin will release O2. But not O2, not all O2 will be used. Like for example, after this vigorous exercise, or we have the oxygen depth, okay, what will happen here? Not all O2 will be used for respiration. It's correct. So this O2 will be released by the hemoglobins, but if you look at the graph for myoglobin, the percentage saturation is so high. So means that myoglobin actually, myoglobin actually will react with O2, will combine with O2, form oxymyoglobin and store this oxygen. Are you clear? So can you see that? So at partial pressure equal to 2, basically our master cells, right? The partial pressure O2 in the master cells. So you can see that adult hemoglobin release O2 at this moment because you can see that the percentage saturation actually lower. But the release of O2, not all of them will be used for the respiration or aerobic respiration. So some of these O2 will combine with myoglobin to form oxymyoglobin. So when oxymyoglobin actually going to release the O2? When the supply is not enough. Let's say, for example, now your muscle carry out a very vigorous activity until the partial pressure O2 in the muscle cells now drop. Okay, to let's say 1 or 0.5 kilopascal. Then you can see that now the supply already, right? There is oxyhemoglobin release already, still not enough. Then myoglobins now release O2. Can you see the drop from roughly about very high 90% drop to about 50%? So means that now myoglobin release the O2. So when myoglobin release O2, so oxymyoglobin only release O2 when supply is low. Are you clear? So this is very, very important. If without myoglobin, what will happen? Supply is too low. It cannot actually sustain already. So in this case, with the presence of myoglobins, we can delay the onset of anaerobic respiration. Are you clear? Can I see that? Okay, it's something like you have the storage, like you have the bank. Okay, you store money in the bank. So it means that if we, for example, like we don't have work anymore, at least we still can actually get the money from the bank that we store before. So it's same like this oxygen, the supply is not enough already. So at least we can go to our bank, these myoglobins to get the oxygen. But if we we delay, okay, the what we call it, let's say no money, you cannot eat anything, right? But now you delay the starvation, it's correct or not? Right, because you still have money you can get from the bank. But now you can see that if let's say without the storage, what will happen? Once it's not enough, the supply is not sufficient, what will happen? Immediately you're going to be starving, right? Okay, so for in this case, we do have the myoglobins act as the O2 storage, release the O2 when the supply is low. Therefore, we can delay the onset of the anaerobic respiration. So means that we won't be able, we still have able to continue our activities. Okay, so this oxygen curve again, as I said, that it lies to the left, okay, of this dark hemoglobin. So they have high affinity. What is their role? They actually act as O2 storage. So they have a capability to extract the O2 from hemoglobins, right? Just now we said that hemoglobin release O2, and then our myoglobin going to take the O2. So they only release O2 when the muscle demand oxygen at higher rate than hemoglobins can supply. Therefore, we can delay the onset of anaerobic respiration. And this is particularly very, very important. Okay, where? In our cardiac muscle. When the cardiac muscle start the anaerobic respirations, the cardiac muscle actually will die because of the accumulations of the lactic acid. But this thing won't happen because in the cardiac muscle, we have a lot of this myoglobin continue to supply this oxygen until no more myoglobins that can actually supply. Okay, so with this, I've done for the fetal hemoglobins and also the myoglobin. You guys have any question, feel free to text, okay, in the chat box now. Thank you.